Okay, thank you everyone for staying until the end and not running away to have your dinner. I'm grateful for Francesca, everyone, to be here and really listen, especially the families, to difficult, perhaps even for us scientists, talks about complex aspects as we move into understand PCDH19. So I'll tell you a little bit about the work of Claire, who is not here, but she was very important to do work on iPS cells. And we do something similar what Jack just talked to you about, but slightly with a different aim or from a different perspective, which I will tell you about. So to start with, I just wanted to remind you that really PCDH19 is quite a multitasker protein. You probably, not everybody can see the bottom bit, but it does a lot of things. It's a cell adhesion molecule. We know also from work of others and polls as well that the protein sits at the synapse. We know that it goes into the nucleus. We have been following the cleavage now for 5 years. We know that the protein interacts with cytoskeleton, and that has been confirmed in multiple studies. We know that it regulates or co-regulates gene expression. That's our work. We haven't seen that confirmed by other studies, but what Sylvia presented is similar. I would like to touch on this because it's an interesting work where actually people found microRNA which regulates PCDH19. And that microRNA sits in a region of a different genetic syndrome which have a lot of similarities with PCDH19 epilepsy. And the second, another knockout mouse was published where there was decreased fear response and the mice were hyperactive, which just confirms what you heard about hyperexcitability, the attention deficit, and a lot of the phenotypes. And from at this meeting we heard about the GABA interaction and cleavage. So there's a lot of data to go through and really try to figure out what is important and what do we target. Okay, so that doesn't work. Also, just a reminder that we have these lot of different mutations and we had to juggle not just the biology but also the DNA variation. But we understand that a little bit better. The other thing is that the hormonal changes are quite important across the lifetime. So we know that the onset is about 8 months. So something really, it's either protective here or maybe not that important. And I will perhaps touch on it. We know that for some individuals there are changes around puberty. For most of them probably towards better, but not for all of them. So again, and we also start getting a bit of a feeling that there might be another change for these women at the time of the menopause. So maybe some of the seizures return. So what we did, we actually focused also on creating iPS cells, these stem cells from skin on the girls. But I'm going to talk to you about the cells we generated from a male. So for the whole purpose of the exercise, all the differences, what I'm going to show you, should be considered as normal. So we see differences between the cells with PCDH19 versus the cells without PCDH19. But both situations we treat our cells, and I would like you to understand it as within the normal range. Because we know these individuals are not affected. So that's the reprogramming. This is the differentiation. The whole reason why we do these stem cells is because we need to have access to brain cells, and that's not possible, obviously. The protocol, what we have been using, it's towards excitatory neurons, and it takes 3 months. So one experiment takes about 3 months to take it from the stem cell to the neuron, and we have done it 5 times. So Claire has been working busily 5 times, 3 months, do these experiments to measure everything that's it's correct. And these are just the different stages, and a lot of what Jack mentioned we see as well, but there are perhaps some differences. We probably need to talk with Jack and reconcile as to how we explain them. And this is just the appearance of the cells and labeling as stem cells go to neurons. Human stem cells as human development are slow, but such is it. We either go for the human cells and take time, or go for mouse cells, but then we just have a different species model. So our idea was, again, generate the stem cells from the wild type and the mutant and see what happens, but also try to do the mixing experiment. What we found as well, as Jack showed you, that there was an interesting stage around the rosettes when the cells become polarized, because before that they are just the cells without their identity, so where is the top or bottom, left or right. But at the rosettes, when they start forming these structures like neural tube during development, they start being different at different ends. And interestingly, at that time, the mutant and the mosaic population really was trying to overexpress the PCDH19. Like the cells were sensing that PCDH19, it's required there, but it was not working. So it was really pushing it. So there must be some feedback telling those cells, we want more and more PCDH19, because it's important here. So these are the structures, and this is how they look under the microscope, and that's somewhat similar to what Jack was showing you. And these are the rosettes at different stages. That's the foci. This is the lumen size, and sometimes out of these rosettes, there are the neurons which are more mature. They start to move away. We see that at the level of the neuronal rosettes, in the cultures without PCDH19, with respect to cultures with PCDH19, there is decreased expression of PAC6 as well as these TBR2, which would suggest that these cells are not the same. That's the whole point of the exercise. They are not the same. So these cells are in a different perhaps stage, or they have slightly different identity. So they seem to be less pluripotent, or they are becoming more mature, so they're perhaps moving more towards the neuron. When we look at these rosettes, they didn't really differ between the wild type and the mutant or the mosaic, and that's measured here across 5 different experiments and multiple rosettes. What we did see that the PCDH19 was required for the neuronal stem cell polarity. So when we look at these structures and these polarized structures, or these foci, so these are like the future rosettes, these are the part of the rosettes, we saw that the numbers of these structures in PCDH19 missing cells, mutant cells, was significantly decreased, as well as in a mosaic. We were also looking at the size of the lumen, and this is similarly what Jack was showing you. This is the large lumen, this is the small one, and this is one which is quite disorganized. And really the size of a lumen is determined whether the cells are dividing this way or they dividing this way, because if it's the asymmetric division, they generate more neurons. So in a way, what we saw mostly like this in a wild type situation, when the PCDH19 was missing, the cells were more running this way, so the size of the lumen was actually decreased, which you can see here. So the polarity and the division was altered. And we also saw a markers of more mature glutamatergic neurons appearing. We also saw accelerated differentiation. That's what Jack was also talking about. So in a way, these cells more push for neurons to be generated rather than proliferating or renewing themselves. But this is all should be considered within normal range. So this is not pathogenic and this is not pathogenic. Here the mosaic really doesn't report on the situation what Paul showed, that there will be difference. We don't know exactly why, but perhaps my interpretation is that this time it probably doesn't matter very much. It will matter only later in the development, and that's why, like Jack or Paul, you need to go to more complex structures of the brain to actually get that phenotypic outcome. We generally didn't see a difference in the neurons, how long the processes were, but we did see some difference in the size of the axon, which is a special protrusion of the neuron, which make connectivity, connects with other neurons, which is also might be just a simple indication that these neurons are more mature or they maturated early. So the model is that perhaps a loss of PCDH19 function results in reduced progenitor pool and leads to accelerated neuronal differentiation. Now we saw this also in mouse, and we published that with Paul in his first knockout paper, where we saw these neuronal stem cells in the knockout mice really moving further away from the stem cells. And this is the wild type situation. You can compare it. And we also found that the differentiation, which was not published yet, is increased towards neurons in the mouse. Now another support comes from that microRNA 484, which regulates PCDH19. Now this microRNA sits in this chromosome region 16, and that region, whether you have duplications or loss of one copy, causes attention deficit hyperactivity disorder, intellectual disabilities, epilepsy, developmental delay, speech delay, autism, behavioral problems. If I cover 16p13.11, it is the same like you were reading a clinical description of PCDH19 related epilepsy. And moreover, there is variable penetrance in males versus females, which is quite intriguing. Now it's interesting that overexpression, because of the duplication, for example, of 484 leads to which is equivalent to the loss of PCDH19, decreases proliferation and increases neuronal progenitor cell differentiation in vivo. That's pretty much exactly what we have seen using the iPS cell model. And the fact of this loss can be rescued by ectopic expression of PCDH19. You may not see it really well from the back, but the microRNA during development, the microRNA expression and PCDH19 expression are sort of inversely complementary. And when you generate a mice which overexpresses this microRNA, they just simply have less PCDH19. So it's a highly relevant model which to some extent validates also what we have seen using our iPS. But what is really interesting is their hypothesis that this leads abnormal neurogenesis, which we would support based on our studies, but also hyperactivity. And that's to some extent a speculation on behalf of those authors. Now, the other thing what we looked at, you remember, and Ewan showed it, that we found oxytocin receptor dysregulation. Now, oxytocin receptor is very important because it's a therapeutic target as well. You know that in autism and other disorders, people trial oxytocin nasal spray. But we also see like glutamate receptor GRIA1 dysregulation. So you see GRIA1 go down in patient and oxytocin receptor goes up. When we look at our cells, we see something similar at the new rosette stage. The GRIA1 is significantly down. While here, oxytocin receptor is not statistically significant, but you see it from rosettes, it suddenly goes up, which is very much similar to what we see. Now, why am I talking about this? Firstly, these cells, I believe, are different from very early development. So these cells carry their identity from very early, perhaps where PCDH19 more function as a regulator of gene expression rather than cell adhesion molecule. and the cells from there onwards are different. Now, oxytocin receptor itself is very important because you know, and interesting, nobody talked about it today, about the GABA switch. So GABA receptors are excitatory or before they become inhibitory. And it is oxytocin receptor which have been recently identified to actually regulate this switch. And I've just told you that PCDH19, and we know it from patients, regulates oxytocin receptor. So it's quite possible that something is happening in this pathway as well during critical phases of development where this switch, it's delayed. It usually happens at birth, at least as animals. I think in humans it might be a little bit later. But the possibility is that this switch from GABA being inhibitory rather than excitatory is delayed or is somehow affected. So during development, early stages, perhaps PCDH19 works as a gene regulator, then it works as a guidance molecule or perhaps sorting molecule, and as Paul showed as well, then later on works at a synapse. And I think if we are going to understand PCDH19, we need to look at this molecule from all these different angles to understand how to manage the disorder at an individual. So to summarize, what I was trying to say is that these cells, positive and negative, are different from early stages. However, we need to look at these differences as within the normal physiological range. But what we do see that the human skin cells, iPS-derived neurons and mouse neurons, there are common themes appearing, which reinforces perhaps we are on the right track. And that PCDH19 may have different function at different types of development. Or some of these different functions can be recapitulated in mature cells if, like Sylvia showed, that the activity-dependent translocation and cleavage of PCDH19 into nucleus and gene regulation. Interestingly and intriguingly, loss of PCDH function might be different in different organisms. We know that the mouse doesn't have a very overt phenotype, certainly nothing like we see in the patients. The question is then, when is also the critical period for intervention? Is it really pre-puberty? Is it within the first year or two of life? Or what time? Because in mouse experiments, in similar epilepsy syndrome, sometimes we need to intervene, let's say, within the first 10 days of their life to achieve possible outcomes. And of course, the question is what to target, positive cells, negative cells, or the secondary effects. Perhaps, like we heard today, to destroy PCDH19 in every cell might be a strategy. But I'm a little bit wary about the efficiency of it. So to me, I look at this epilepsy as a disorder of coexistence of two different but otherwise physiological states of neurons. And as such, I'm looking at it as a possibility of treatments. We are not really treating the disease. We are adjusting two normal states back into some other state, which is not going to cause problems. So that's where I'll stop, and thank you all for your patient listening. Thank you.
Okay, thank you. So now the two talks are open to discussion, and Jack, can you come back? Thank you very much. Some questions?
Yes, Joseph, I was very intrigued by the idea about the GABA switch. Is that something that do you think that that happens also postnatally? The reason why I ask that question is if there is this GABA switch, is it possible that by giving GABAergic drugs, you can actually cause an exacerbation of seizures, or is that purely a prenatal phenomenon?
Yes, now I can see it. To be honest, I really don't know a lot. This paper was published, Jack could probably comment on it, only very recently, and we have been interested in oxytocin receptor for some time because it was one of the most valuable markers in patient skin cells, where we significantly, it is estrogen receptor target as well. It's expressed in a sexually biased manner, so we have different levels of oxytocin receptor in males and females. So everything pointing towards important function. The oxytocin of knockout mice have behavioral phenotype. Oxytocin overexpression has been found as well, receptor expression. So the switch was really a very interesting finding, and as I understand, it's happening postnatally, and perhaps here might be delayed.
Yeah. Okay. Yeah. So in rodents, it's usually, it varies by region of the CNS, but usually by the end of second postnatal week, the switch is complete, and it can, for example, in adults after status, there's some evidence that it goes back to the embryonic state, and it prompted a trial with bumetanide, which influences NKCC1 and KCC2, but I don't think the results were positive. I'm sure some of the pediatric epileptologists can speak more to that. So it is a theoretical issue, but it's harder to show in vivo.
Recently, we have published the first demonstration of measurement of chloride concentration in vivo by two-photon imaging. And what it turned out is actually quite interesting, and it's something we are following up, which I think it might be important for many pathologies, not just PCDH19, is that yes, we do see the shift during, as has been described by electrophysiological methods mainly, during development, but we find that there is always a population of high chloride cells, even in the adult. And there is a period of development where actually you see a mosaicism in chloride concentration, and the cells that carries high chloride concentration responds better to bumetanide. But that never dies down. The notion that chloride is 5 millimolars in all adult neurons 24 hours a day, always, I think is not correct. I think it's more complicated than that. I think there is an active regulation of chloride concentration in neurons, and I think this has tremendous, tremendous opportunities in terms of explaining mechanism of pathology. So I think this is something should be followed up. Is quite interesting.
Yep, thank you. Dina.
A question for both of you. How can we explain that the depletion of PCDH19 stimulate the differentiation of neurons? Have you some hypothesis?
Well, based on what we think, it's simply the polarity, because PCDH19 interacts directly with N-cadherin to form a specific interaction interface. And that interface might be slightly imbalanced by loss of PCDH19 or loss of function of PCDH19, and they can maybe slightly push the cells from the symmetric division to asymmetric division, and as such the cells perhaps go into the differentiation a little bit earlier, which overall it's not a major problem like in a male. So the shift or all of that change is probably relatively minor, that it doesn't matter.
And a second question, from a clinical point of view, do you think that the iPS-derived neurons can be a useful tool to functionally validate the missense mutations in patients? Or it's too time-consuming, or?
IPS are very expensive. It probably would be quite time-consuming, and we seem to have better methods which are moving towards in silico only prediction tools which are going to work perhaps on PCDH19 very successfully.
Even the rosette, because you have a 15-day culture?
I probably wouldn't go to individual mutations unless you find something very interesting with a different phenotype, maybe different part of the PCDH19 protein.
Yeah, so
Jack.
I mean, we're actually trying to put together a grant to look at that, comparing different model systems, including zebrafish, iPS cells, and rodent, including in utero electroporation for rodent. And I think for different genes, it all comes down to the assay that you have and the phenotype that you're looking at. There are ways to pretty efficiently get genes into the safe harbor locus to express it, say on a null background. But again, these aren't easy, as Joseph said. It's time consuming.
Yeah, thank you very much.
Thank you.
Mi scusi, un'ultima considerazione. Volevo fare una domanda così, forse sembrerà un pochettino un po' anche sciocca, nel senso, per capire cosa avviene quando mia figlia, quando gli avvengono le crisi, va in coma farmacologico, e questo ci sta anche diversi giorni in rianimazione. Si può fare qualcosa per questi eventi così acuti? Cioè, perché lei ogni volta che prende le crisi si fa il buccolam, sembra che rinviene, poi dopo ritorna nel post critico, assenze, midriasi, tonico-cloniche generalizzate, tutto quello che volete, però lei non riprende il suo stato di coscienza, ed è così per giorni, finché non desatura e viene intubata. Vorrei sapere cosa avviene, cioè si può fare un qualcosa al riguardo.
I guess I have the MD, so maybe I should talk. I mean, so those are always very difficult situations, and you're dealing with the effects of seizures plus the effects of all the drugs that have been given to stop the seizures. And, you know, for the most part, it's hard to show, other than with convulsive status epilepticus, that seizures do severe damage to the brain each time you have a status epilepticus episode, but with focal seizures, you can affect memory areas, and so patients often say, you know, they come back, but not quite back to their baseline in terms of learning and memory and those issues. And we don't really have agents that protect the brain that we can give during those times. There's been a lot of studies looking for those, but we haven't achieved that yet.
My area of expertise to comment. I would need to ask clinical colleagues from the audience to reflect on it. Carla, yeah.
Like perhaps I'll do it in Italian, yeah. Alla signora che ovviamente salve, ci sono appunto dei pazienti che hanno degli stati di male che magari si prolungano per un po' più di tempo, quindi hanno bisogno di un trattamento più invasivo, un pochino più prolungato. Poi la risposta ai farmaci rimane comunque abbastanza individuale, probabilmente anche geneticamente determinata. Quindi è possibile che certi farmaci su sua figlia abbiano un effetto un pochino più importante, magari deprimono il respiro, per cui viene intubata per una questione di sicurezza, quindi a volte è una questione di dose, anche di severità e di appunto di che tipo di stato di male è. A volte, se è uno stato di male non convulsivo, ci permettiamo di fare un trattamento non essendo convulsivo magari di non essere così pesanti con la terapia, quando invece diventa uno stato di male convulsivo allora siamo anche più aggressivi come terapia, proprio perché pensiamo che comunque uno stato di male convulsivo possa danneggiare ulteriormente i neuroni, quindi non vogliamo che duri più a lungo, per cui magari il trattamento è un pochino più pesante e questo può richiedere un'intubazione che è sempre solo per mettere in sicurezza il paziente. Ok. I wanted to make a comment now that I've got the microphone to these presentations. Now that we probably understand a little bit better the mechanism of action of PCDH19, probably we should look at the MRI of these patients more carefully, and because it's possible then they might have some structural abnormalities that we have overlooked over time, thinking that the MRI was actually normal, but maybe there is something we have not paid so much attention so far, and possibly maybe this focal dysplasia are actually not that uncommon, or if not focal dysplasia, maybe some sort of structural general abnormalities. So that was just, maybe you want to make a comment on that, and maybe we should do some different imaging of the brain, perhaps PET studies or some different studies to, I don't know, see if there is really, the brain is really like even structurally normal.
Yeah, no, I think what Paul showed was very interesting, and the recent publications. I was going to ask you, is anyone doing DTI?
We have not done it so far, but I think we should really
That
do
might
it.
be
We should look at
interesting,
the MRI. We should do DTI. We should look at the DTI, and we should look at the MRI in a more careful way. Yeah.
Yeah, we looked at many MRIs. Francesca kindly also shared with us maybe 15, 20 different patients together with my colleagues in Melbourne who are experts in it, and that's how we found some of those cases which sort of not really focal cortical dysplasia but unusual MRIs which Paul showed, but it was really very hard to put a finger on it exactly what it was, and it was definitely not consistent across patients. Certainly the tools for looking at the connectivity are probably the more exciting ones we could imagine, but it would be good to have some traces for GABA, perhaps that's what we considered, like, or even oxytocin receptor, as I understand, very soon can be able to be imaged, but perhaps if we started with just GABA, they may give us some answers.
That's why perhaps the PET studies could be using a GABA tracer.
Yeah. No, we try to. Sometimes it's not that easy to get the girls back into the magnets for those investigations.
I had a comment about that, and then another question for the presenters. But at my university, we had an unusual a case of an adult woman who had focal epilepsy for her whole life and was found to have a dysplasia on MR and PET hypometabolism in that area, who was operated on and had a pathologically proven focal cortical dysplasia. And after her surgery became seizure-free, and then subsequently her niece developed epilepsy, multifocal epilepsy, and was diagnosed with PCDH19. And the woman, after her surgery, then underwent genetic testing, and she was also found to be positive. So I think in many instances, the children that I take care of and that we take care of have multifocal seizures, and it sort of precludes or obviates the desire for the clinician and the family to undergo more advanced imaging, but it's an important question. And I had a question for the panelists about looking at, in your models or in your experience with stem cells, any comments about future directions to understand the nature of the seizures to cluster? It seems just so unique to this condition. What do you think we can learn from from stem cells about what the reason is for that, and would that potentially offer other treatments, like this idea of the GABA switch and involving oxytocin receptor? Is this dynamic, and does this make the seizure clusters persist for days and days, which is just unique in this condition compared to others? So what can we learn about the clustering from the models that you're working on?
I think with Jack, we haven't really discussed it, are we converging on the same I guess, technology, at least for the time being, to look the MEAs, or use the MEAs for specific types of behaviors of these cells or slices from the mice, because we can have not just cells but also slices, and to compare the activity to wild-type regions and compare the impact of known and new drugs. So it would be really very interesting to see how those experiments go. We're now optimizing our MEAs for exactly those purposes, to see how these cells behave and compare it to existing epilepsy models using that technology, because we have access to SCN1A through our colleagues, which is the Dravet syndrome, SCN2A, which is very well known cause of different epilepsies, autisms, and neurobehavioral and developmental problems. So I guess that's perhaps where the advantage will come, looking across different disorders and see what are the similarities or differences. The bursting, I don't know. The connection to NONO, it's very intriguing, because NONO is involved in circadian rhythm regulation, because it's a part of the group of proteins which regulate circadian rhythm. I'm more actually intrigued by the temperature control and relationship to temperature stresses and how that feeds back. So there must be some kind of loop which predispose these clusters of seizures rather than have intermittent.
Yeah, I'll just add, I think that's one of the weaknesses of the stem cell models, is when you really want the nuts and bolts of the network activity, you have to go in vivo to look at things like seizure clustering and things like that. So hopefully some of the mouse models will start having spontaneous seizures, and we'll figure that out.
You believe in mouse models. You believe in mouse models. I probably don't anymore.
Allora, dobbiamo finirla qui, perché intanto le povere interpreti sono distrutte, quindi non possiamo abusare ancora di loro. Ringrazio tutti e solo i ricercatori domani mattina qui alle 10 per i working group. Invece stasera siamo tutti invitati a questa bellissima cena offerta da un'associazione di chef che si chiama Con il cuore nel piatto. La cena è alle 8:30, io non ho dato l'indirizzo, quindi ve lo do adesso. Largo Santa Lucia Filippini numero 20, vicino Largo Argentina, al centro. Lo ripeto, Largo Santa Lucia Filippini numero 20. E anche coloro che non avevo invitato per email ma che sono qui adesso sono invitati, quindi scusatemi, ma abbiamo avuto tanti problemi organizzativi in questi giorni. Va bene, allora a fra poco con tutti, spero, e grazie insomma a tutti per aver partecipato.